Harvard Divinity School.
Planta Sapiens and Human Impatience. Are we patient enough to learn how smart plants are? April 11th, 2024.
Good afternoon, good morning, good evening, depending upon where you're joining us from. My name is Rachel Peterson. I'm a third-year MDiv, and on behalf of the Center of the Study of World Religions at Harvard Divinity School, it's my great pleasure to welcome you to our conversation today with Professor Paco Calvo, titled Planta Sapiens and Human Impatience, Are We Patient Enough to Learn How Smart Plants Are? This talk today is sponsored by our Thinking with Plants and Fungi initiative, which explores how inquiry into plants and fungal life illuminate the nature of mind and matter and humans' relationship to the more than human world. We engage fundamental questions arising from academic scholarship as well as traditional wisdom, including what is intelligence? Where does it extend and how? What is matter and what does it mean to call it animate or inanimate? And how can we broaden our understanding of care to include other species and other forms of life? We are very grateful for the V.K. Rasmussen Foundation for their generous support. And in case you weren't aware, we offer several opportunities, including a bi-weekly reading group, a guest speaker series of which this is a part, experiential field trips, and regularly scholarly content. So if you'd like to learn more about that, please sign up to the newsletter via the CSWR website. And with that, I'm going to pass it off to my colleague, Natalia Schween, who will introduce Paco and our talk today.
Thank you so much, Rachel. Hi, everyone. Thank you for joining us today. My name is Natalia Schween, and I'm a second-year PhD student in the Committee for the Study of Religion. We're so excited to be doing this today, so thank you so much for joining us. So a bit about what today's talk is going to be about. Plants have long been deemed passive organisms with hardwired or inflexible behavior. However, a growing body of empirical research reveals that plants exhibit cognitive capabilities traditionally attributed to animals. And yet controversies over these scientific findings have recently intensified. Professor Paco Calvo joins us today to speak on Planta Sapiens and Human Intelligence. Are we patient enough to learn how smart plants are? Paco is a renowned cognitive scientist and philosopher of biology, known for his groundbreaking research in the fields of plant cognition and intelligence. He is a professor at the University of Murcia in Spain, where he leads the Minimal Intelligence Lab, focusing on the study of minimal cognition in plants. Calvo's interdisciplinary work combines insights from biology, philosophy, and cognitive science to explore the fascinating worlds of plant behavior, decision-making, and problem-solving. Today, Paco will reflect on the current challenges faced in the fields of plant signaling and behavior, including risks of underdelivering and strategies to avoid biases that may lead to overinterpreting results. This talk aims to spark a multidisciplinary dialogue around the questions of plant cognition and event sentience, and to foster renewed scientific curiosity into the rich cognitive landscape of our green companions. Rachel, any thoughts before we begin?
That was perfect. Thank you, Natalia. Just a brief comment on how this event will go. Professor Calvo will present his presentation, and then Natalia and I will reappear to engage in a few questions for our friend Paco Calvo. We'll pass it over to you, Professor Calvo.
Yeah, thank you, Natalia, Rachel. Yeah, thanks a lot. Thanks for having me and everybody here for the interest in plants, right? Plants are what we want to talk about. Well, actually, Rachel, Natalia, and the rest, I do have a question for you, right? It's in the very subtitle, right? Are we patient enough, as you said, to learn how smart plants are? And I was thinking, if this was like a 1-minute talk, the answer would be no. Thanks. Any questions? We are definitely not smart enough. I mean, there are so many problems, so many challenges, and we can do way better, right? So rather than talking about the success in the field, although I should say a little bit about that as well, let's see what lies ahead. If we can see what can we do better in the understanding or in the scientific study of plant intelligence and sentience, right? And if that is the case, I mean, if the answer is no, we are not smart enough, there is a lot of work to be done, then the question is, well, why are you so smiley with Natalie Lawrence here? We are, oh yeah, sure, Planta Sapiens. Plants are intelligent, they are sapient. We mean it literally, right? So that's the idea of the very title of the book. We say, no, this is no metaphor. We mean to say that they are sapient in the very same way that Homo sapiens means humans are intelligent, right? So we mean to say this is not a stylistic resource, right? In fact, there are a lot of reasons to be happy about or to be optimistic about, like the interest in the field. In our case, it's been translated to many languages, the message is being conveyed, it's being passed, and we have the feeling that the topic is catching on and people are starting to to be aware that yeah, sure, I mean, plants are not just, they are not objects sitting there being rooted doing nothing. I mean, they've got to be doing something really smart, otherwise they wouldn't be here, right? And yet, when you see the type of coverage that, at least in our case, we've had in the last few years, you see those covers and there is something funny about them, right? I don't even need to say what it is, but do we need to portray a plant intelligence as a thinker, like rodents thinker, or they need to be brainy? Do they need to have brains somehow to be able to show their smarts? Looks like we are, in a sense, not quite getting the idea of what plant intelligence is about, right? So we are basically anthropomorphizing them. We don't quite get it, right? We are so obsessed with ourselves that this is this navel gazing attitude that we have, that we think, oh, if they are smart, to whichever extent they are smart, they've got to resemble us somehow, right? We are like the gold standard. And there is a lot to be said about that. There are a lot of skeptics in the field, insiders and outsiders, that they say, no way, I mean, you just don't get it. Plants are not, they cannot be intentional beings. Yeah, sure, they grow, they develop, they do their things, they exhibit adaptive behavior, but that's not intelligent behavior, that's not cognition. Plants cannot be sentient, right? And today, what I would like to do is not to talk about the skeptical attitude of those outsiders or insiders who just cannot quite understand it. Doesn't matter how much you explain to them or how much literature you try them to read or to inform people about the scientific literature in the field. They just wouldn't get it. They wouldn't buy the message, the idea. That's not what worries me. What worries me is what we are missing from the inside, what we in the field are missing, what we still don't get, right? So there is this big elephant in the room in the plant sciences, and we are not being sufficiently self-critical, I would say, right? So it's very easy to simply blame, current culture of fast science and say, yeah, sure, I mean, this is the problem we all have, the academic inflation, publish or perish, or even worse, doctoral students now, the joke, right? Publish, then perish. So those two jokes, I don't know which is worse. Well, the second, right? But it's not about that. It's not about this culture of fast science. We know that doing slow science is the name of the game. We know we've got to chill out, to slow down if we want to do something, a genuine contribution to the field, right? But that's something that affects the intellectual academic culture in any field whatsoever. So we want to see what's the specific, what's happening within the field of plant intelligence or plant sentience studies that is preventing us from making more steady progress, right? And in a sense, if you think about it, we cannot speed things up if we put it in terms of the disciplines involved, mainly because we know a lot about plant physiology. You have the nuts and bolts. Well, of course, it's a lot to be known, but we have a decent amount of solid foundations when it comes to the biochemistry, to the molecular cellular biology, to the plant physiology as such. So we know a lot, but we forget that we cannot approach plant intelligence from a mechanistic or a reductionist perspective, right? So we certainly need to connect the dots with the upper level disciplines, and we need to include all the insights from ecology, and be speaking of ecophysiology, and not just plant physiology, but plant ecophysiology. We need to be speaking of the evolutionary and developmental biology constraints that allow us to understand what a plant is up to, both in ontogeny and in phylogeny, right? What is it that they are up to? Otherwise, we wouldn't make sense of the idea of plant learning if we didn't think in terms of ontogeny, right? So all these ideas allow us to say, hey, we cannot speed things up. We need to talk to the different subfields, see how to bridge, to establish connections in between them, and then we might be able to, to raise a different set of questions. Now, you know what the problem is? That this is, in a sense, pretty obvious, right? So anyone would say nowadays, yeah, sure. I mean, this is emergent behaviors, properties that defy a reductionist understanding. I think we are all on the same page here. But what we are missing is that doing this effort, strictly speaking, from within the plant sciences as such, reductionistly or not, mechanistically or not, just cannot deliver the goods. Because we need to be speaking about the cognitive sciences effort. We need to team up with the cognitive scientists. So when you saw in the previous slide that upper little vertex where I put the label philosophy, in truth, what I mean to say is that one vertex of this hexagon should be not just the philosophy, but the cognitive sciences as such. So the very same effort we apply when we say, hey, let's do animal, comparative psychology or animal cognitive neuroscience. So when we are thinking of importing or applying the tools from human psychology to study the cognitive capacities of non-human animals, right? When we are doing that, we need to understand that those two have to match or to work truly collaboratively. Otherwise, we would be doing the cognitive sciences and the plant sciences, and we would still be missing the plot. We wouldn't be raising the right sort of questions, right? So this is what we are after. We are after something which is like not the cognitive hexagon, not the plant hexagon, but the plant cognitive science hexagon. So putting all this together, right? So that's in a sense what we are trying to do at the Minimal Intelligence Lab here in Murcia, Southeast Spain, if you want to know where we are based. And truly, we mean to say that it goes beyond an interdisciplinary or a multidisciplinary effort. So it's got to be truly transdisciplinary. And this includes, by the way, the arts and the humanities as such. There is a lot to be said on that front. Maybe we can discuss that during Q&A, but I won't say much now. But this is not just the plant and the cognitive sciences. This collaborative effort, in our case, this is the home crew here at MintLab, and just the latest, for example, Carla Cao, bottom left, just joined us a couple of months ago, coming from Florida, from Miami. And look, yeah, plant phytoacoustics. Okay, we want to know about, music cognition. So we need to bring those backgrounds, right? Otherwise, we would be speaking, or we would be risking speaking about plant or phytoacoustics, the recent field of plant acoustics, in a metaphorical way without getting to understand exactly what we are after. No, we are after the very same thing that we do when we wonder whether animals perceive sounds the way they do it, and etc. Right? So we are simply applying the same protocols, the same methodologies, the same principles, theoretically speaking, to non-neural subjects, but subjects after all, right? So that's what we do, and we have two ways to peek into the plant, from the inside out and from the outside in, and we have to put them together. So we can take take pictures, assemble time-lapse footage, and yeah, sure, you watch a clip. Okay, now I see the plant in motion. But you can also insert electrodes and check what's going within the plant, inside the plant. So you can just do recordings of electrophysiological activity and see how those correlate. What's the name of the game when you do that in animal cognitive neuroscience? You might say, hey, I'm wondering about the neural correlates of some, cognitive capacity, or the neural correlates of consciousness, right? Well, literally, we are doing the same. We are after the non-neural correlates of some plant potential capacity that we are trying to identify. See what I mean? So it's not that different after all. In fact, if you think about it, things are way simpler. We make things more complicated because we don't quite get that the rules of the game are the same. If only we need to customize the protocols, the methodologies, to make them applicable in principle to a subject, plants, that happen to exhibit their behaviors way slower. So it's the time scale what counts and what prevents us from appreciating their doings, right? Other than that, the rest remains the same. And that's why you can put the two hexagons together, the plant and the cognitive science hexagons, right? So when we do that, for example, we can ask whether a climbing plant, in this case, a climbing being, is controlling the way it's approaching a support. Is it simply growing and developing and out of mere growth bumps onto the pole? Or is it somehow controlling the approaching maneuver, right? So we can do that in the lab. We can, I mean, I insist, kinematically, dynamically, in the very same way you would ask whether a non-human animal is controlling the way it's approaching a target. Or is it decelerating? Is it accelerating? Is it bumping onto it? Is it controlling the way it's approaching? You see, velocities, accelerations. You can play the same game of unearthing what's going on within the system, within the model, by inferring it from cues you can track, right? So that's just an example of what you could be doing. Now, wondering whether a plant performs a goal-directed activity in a flexible manner, or simply, as opposed to simply bumping onto it and say, oh, lucky me, I got there, right? No, I'm controlling the way to get there. There is an issue here, because we need to be careful when we watch clips. Watch this clip from the 40s. This is amazing. I'll let you watch it, okay? I'll just let you watch it. Okay, he didn't quite like it. Didn't you see something? I mean, raise your hand if you were able to see it without anthropomorphizing circles and triangles moving around on the screen. You just couldn't. There is no way you can do that. Okay? So it is impossible. It comes with our psychology. We cannot help it, right? Now, what happens when you watch the clip on the right? David Attenborough's classic. Well, now we see plant growth. We've done the very same thing. We speed them up like in the lab. We see what they are up to. Oh, look at that. Wow, wow. As if feeling for the best way forward. I don't know if you can hear David Attenborough's soundtrack. Can you hear his voice? No, you cannot. Okay, that doesn't matter. So David Attenborough is saying they are going as if they were reaching for effortfully, as if they are trying to, as if. Okay, so you see what I mean? How do you get to know whether look at that? Oh, there it goes. Yes, I'm making it. After all this effort, it takes time for me to grow, but there you go. Look, out there, all the way up, reaching for the sunlight. That cannot be coincidence. They are they know what they are doing, right? We would like to think. Now the question is, how do you know when you are actually witnessing genuine plant behavior as opposed to inflating triangles and circles being moved around unintentionally? Think about it. You weren't able to see the triangles and the circles as geometric shapes. We just couldn't. Why is it not the same? I mean, is there not a risk that we are overinterpreting the time-lapse footage and saying, hey, look at that, plant intentionality. I can see it. I see plant intentionality. Well, if you think you are seeing plant intentionality, then we have a problem. You cannot see it. All you can do is infer it, right? So we might see some pattern of behavior whose complexity, the degree of sophistication, flexibility, whether it's anticipatory or not, might allow you to guess that, oh, there might be some meaning making in the backstage, right? But you cannot see it in the very same way that you cannot see the intentionality in the triangles and the circles. Now, so this is something we need to be careful about, because remember, my worry is not the skeptics, my worry is ourselves. So those that, when I say Planta Sapiens, they say they nod. I say, hey, don't nod. Don't buy the message before I explain you why they are intelligent. So we've got to be careful. Say, hey, don't overinterpret the data. First, let's have solid data, solid foundations, a solid theoretical framework. Then we can understand why we get there. But not because we want them to be intelligent. We need to understand why they are so, if they happen to be, right? So all these biases we fall prey on a regular basis. Another bias we fall prey of is the IKEA bias. I don't know in the States, but here, you know, you buy this IKEA furniture. This is like this furniture you assemble, like the Swedish company. So you assemble the furniture, and you know it was so effortful because you had to follow instructions, but you skip them, and then you skip a few steps, and then you were doing this, doing that with the key, the Allen key, the screwdriver, and then the table is not that well assembled, but it's your table. You made it. You are sweating. It took you 2 hours. You love it. So when we put a lot of energy into something, and that's what we do at MintLab, and of course everybody everywhere else, we are DIYing. And think about it. Why do we need to DIY our equipments? Because they are usually thought for animal models. So that's one of the things we need to do. We need to customize those equipments for plants. Different time scales, different spatial scales, different everything. But if we are going to unearth and appreciate plant sophisticated over behaviors and the underlying electrophysiology with the electrodes, then we need to think of, oh, this is just something we were using some wooden scaffolding to allow for the electrodes to be inserted without making noise onto the stem, blah, blah, blah. So we are assembling IKEA furniture. And in doing so, we need to be careful not to fall prey of that effect, because we might be so in love with our work in the lab that we might be rushing our interpretations. So extra caution is needed, right? Well, take this case. I mean, there is a case in which, I'm not going to make a spoiler. Let's see if you see it. This is it was breaking news in 2016. Plants can learn like Pavlov's dog, right? So like they can perform associative learning. So basically, we know, of course, they would grow towards blue light, and that's you don't need to condition them to do so. They just by nature they would go towards phototropically. They wouldn't care for an airflow produced by a fan or something, but when you pair those two, the fan and the blue light, they would we would be able to trigger the conditioned response, right? So in the presence of the airflow, if they have got the association, they would say, hey, I'm going to grow this way because the blue light is going to come next, right? So that's you see, this is a clear example of thinking in animal terms and then importing the protocol into the plant model and thinking of what to do. Now, the bad news is that all the literature in plant learning and conditioning, 20th century, hasn't been replicated. Remember, fast science. So we have a lot of anecdotal results. And you don't know if this is a false positive. You don't know if this is a false negative. We don't know because they haven't been replicated. So this is a serious issue. You see what I meant? So let's not put too much cart before the horse. When you do that, you say, okay, let's think about the experiment itself. And you see, we have the pea plant, we have the Y maze. It can grow onto the left or the right arm. And then you see, now you understand the rationale and the methodology, right? So, okay, if they are growing towards the fang, okay, then fang light, okay, fang, I would grow left or I would grow right? Anything you find awkward about, so imagine, so you train the plant for 3 days, and the 4th day, the test day, you walk into the growing chamber and check where it went, where the pea plant grew into. Okay, did it go into the left or the right arm, right? You peek through the top of the Y maze to see where it went through, right? Anything you find fancy about it? Feel free to respond. I don't know if they can on the chat. Maybe I shouldn't open this. Okay, so I'm gonna tell you. Otherwise, but basically, that we are not seeing plant behavior. This is an opaque Y maze. You don't see what's going on during the experiment. I want to see the pea plant growing. You don't see it. You wait for the test day and say, hey, let's see where it did go into. But you don't get to see it. So if you open a little window, which we did, of course, this is just out of curiosity, I couldn't help it. I just broke the Y maze open and say, hey, let's see what's going on here. Of course, that wouldn't work because you would have light coming from the front. But once you do it in controlled conditions, you get to see something like this. Okay, watch this out. I'm going to speed it up because this is slow. Okay, this is the pea plant growing. Okay, now this is a dark chamber with infrared light so that we can record in pitch black. Now you see the pea plant growing into the Y maze. It can go left, it can go right. And look, it went straight in the middle onto the plumb line, stuck onto the bifurcation in between the 2 arms, but keeps growing. So it's pushing because it keeps growing. Eventually, oops, it will let itself into 1 arm. You see? Now imagine you had seen this at the very end without being able to watch the actual behavior. And then you walk into the chamber on the last day and see, hey, what you see right now, hey, it grew onto the left. And they say, yes, plant associated fang and light. Well, now you see what actually happened. It was stuck in the very middle, growing vertically, and eventually it let itself go out of mere physical forces. You see? We cannot overinterpret the data. We've got to be careful. And this example is great because think about it. With plants, we don't think the very same way we would with animals. Say you are doing an experiment with rats, lab rats, in a maze, and you put the cheese at the end of the maze. And would you ignore the way the rat navigated the maze to get to the cheese? You want to see what the rat is doing, right? You don't want to go on the last day and say, hey, is the cheese there or did it eat the cheese? You want to see the actual navigation pattern, right? But we haven't been questioning that about the plant case, right? So we went through a very careful study, and it took us 2+ years to accomplish this. We call it the 11 issues paper. And it's just a paper on, well, I won't go through this, don't worry. But if you browse it, you will see that we list all the important, substantial topics that were being ignored, that were being turning a blind eye in the discussion on the possibility of plant learning by association. So then you get to think, what's going on here? Fast science. Are we speeding things up? Are we taking enough time to mull over the results and think carefully and do the controls and the pilots that are needed, right? So it's a lot to think about. By the way, if you think about it, the very title of the book, Planta Sapiens, you can guess that I'm not suggesting that plants are not able to learn. In fact, my guess is that they are. We just need to have the solid data that backs up the empirical hypothesis, right? Now, once you do it, once you have the solid data, you have another problem. right? I'm going to tell you a lot of problems, right? So what's the next problem? Next problem is that, oh, yeah, this is a cuscuta growing onto a host plant, because cuscuta hasn't got chlorophyll, cannot do photosynthesis. So it's a parasitic plant. It needs to suck up the nutrients from a host plant. It's doing pretty well. But you might say, oh, what's the big deal? I mean, even if this was not simply bumping onto the plant, but actually targeting it and going for it, someone might say, well, but it's reactive behavior. It's simply reacting to some sensory information, right? Well, not quite, because we can see the behavior actually as the observable consequences of the choices cuscuta is making. So that's the one in the middle is a cuscuta that is doing pretty well. You can see how it's covering the host tree, right? Cuscuta, if it's giving a choice in between a more juicy host and a more poor host like wheat, it will go for the juicy one and not for the poor wheat without energy. But if there is no juicy hosts around, it will go for the less juicy ones. So they will, in a context-dependent manner, choose one course of action or another, depending on the local conditions and the situation, right? So it's not simply reacting to something. It's got to plan ahead which way to grow into anticipatorily, right? Of course, there is a whole story as to how that gets done. Again, that's something we can go through in the discussion if you're interested, but just to give you an idea, it gets pretty sophisticated because it's not simply responding to some cue. So it's not just light related, or it's not just, VOC or volatile related, organic compounds that they can sniff, or sound, by the way. Remember my student working in plant acoustics. Sound might be part of the equation. We have to speak of the integration of all those cues. They are not simply reacting to one cue. They are reacting to a whole panoply of cues from the inside and from the outside. So a rich repertoire of informational channels are being processed and integrated in real time. And when we put it in those terms, now we understand why we are speaking of plant cognitive sciences. The vascular system of the plant is not anymore just the way a higher plant has to translocate substances like nutrients, water, sugars. No, it's an information processing network. So the plant is processing information, it's making decisions, it's picking choices. You see? And it's doing it systemically. So it's not simply one tip, one root shoot, one shoot, one root tip, doing its own business, unaware of what's happening at the other end of the plant. No, it has to integrate all the information coming from the rest of the plant to provide a response that is globally adaptive, right? So that's something that starts to look like cognitive or intelligent behavior. In fact, look, I won't go through this. This is just to give you an idea of how long the list can be. We've been compiling, and this is work in progress. This is never ending, because the more you dig in, the more you find. They will be caring about many, 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 many different things. So they are performing exteroception. They perceive the external world, but also interoception, also proprioception. So all these informational channels from the outside and from the inside have to be put together to the service of those global responses, right? Then once that takes place, then at last we can understand that, hey, I know you weren't talking metaphorically. Yeah, they are in the process of, or in the business of making their own decisions. They recognize their king versus their non-king. They are sensitive to risks and behave one way or another depending on those risks, how dire the situation is. They behave anticipatorily, so proactively and not merely reactively, okay? Hopefully we'll be able to show soon, I mean, this is work in progress, once we have those experiments well controlled, that they are able to learn, we have good theoretical reasons to believe they are, etc., etc., etc. See what I mean? So now we know we are talking about plant intelligence. We just need to understand how to avoid falling prey of confirmation biases and all sorts of biases, and avoid anthropomorphizing them, to be able to do it in a genuine, serious way, right? So when you do serious science, the problem we have is that many people say, yeah, sure, let's do serious science within the animal kingdom, of course, because asking whether an organism can feel pain, if you ask that question outside the animal kingdom, people think you are a crackpot. And they don't realize that scientifically speaking, the question is equally serious because it's a scientific question, right? To be tested in the lab or in the open, but it's the very same question. Again, don't worry, we don't need to read this, just to give you an idea of the type of criteria that when we are talking about crabs and lobsters, people have in mind. They say, hey, if crabs and lobsters are able to, to possess, if they possess receptors for the purpose of nociception, if they are able to perform sensory integration, if they respond to noxious stimulus, if they perform associative learning, you see all this. Look, if the animal possesses an endogenous neurotransmitter system, you will see why I mentioned this explicitly. But just look. Okay, you see what I just did? So this is the problem. We are even unable to lay the foundations for this question to be raised scientifically on neutral grounds. Why do you need to stick the word animal into every marker? Why don't you say the organism possesses? The organism possesses endogenous neurotransmitters. The organism shows motivational trade-offs. Why don't you just speak about organisms? And then we will find out whether a non-neural organism is able or not. But don't fall prey of prejudices. Don't prejudge the very question we want to unearth. Because once you start to do so, you realize, once you don't fall prey of those prejudices, you realize, hey, analgesia, anesthetics, oh, all these plants can be anesthetized with the very same molecule we use to anesthetize animals, the very same molecule. So mimosa, you don't need to do time lapse, you can watch it to the naked eye. It falls when you touch it. But if you anesthetize mimosa, look at what happens when you anesthetize mimosa. You touch it, it won't respond, bottom left. So just nothing. Now think about it. What happens when an animal comes out of anesthesia? Well, when an animal comes out of anesthesia, oops, sorry. I want you to see the third one, bottom right. Now it's starting to fold, if only more slowly. It's taking more time, right? It's saying, hey, this is gonna, because I'm still like, I'm coming out of anesthesia. Hey, what's happening? I'm sort of still dazed and confused. You see what I mean? So the fact that a non-neural system can be anesthetized with the very same molecule that we use, and the fact that that was a marker, is what tells us that we were not being careful enough in thinking the type of organisms or what sort of kingdoms, organisms belonging to which kingdoms can play the game, or can this question be raised for, right? Now, when you put it in electrophysiological, from a electrophysiological perspective, you see that it shouldn't be breaking news. Plant cells fire action potentials the very same way animal neurons do. You just change the type of elements and the channels involved, but the electrophysiological profile is exactly the same. You see? So after all, if we know that the anesthetic is preventing an action potential from firing, that's what's happening in the animal. So neurons don't fire because of the anesthetics, right? So now we understand that if we hadn't been so zoo-centric, so animal-centric, we would realize that, oh yeah, sure, the very same molecule is preventing plant cells from firing because they do fire action potentials, right? In fact, remember what we said before, possessing neurotransmitters. Well, plants have all these neurotransmitters. Glutamate and GABA play a signaling role in plants, right? And this is funny because you see, we know that glutamate, the neurotransmitter, is triggering long-distance calcium-based signaling for the purpose of defending against herbivores. So if you take this plant and now you're going to see a caterpillar. It's going to be fast. You can watch a caterpillar coming from the bottom. It will start munching onto the leaf, and you will see how the signal, the calcium wave, spreads all over the plant. You see, boom. Now, if you do that the same, applying exogenous glutamate, you will see the same profile, the same spread of activation throughout the whole plant. You see? Now, this is funny because when this happens in plants, the skeptic doesn't like the talk in terms of neurotransmitters. Say, no, but it is a signaling role. We see the spread of activation and how the plant is starting to prepare to defend at other parts of the plant body against the future attack of the herbivore. And we don't want to see it because why? You see? You know why? This is imprint. So people have responded to this imprint saying, no, glutamate or GABA cannot possibly be neurotransmitters in plants because if you go and look it up in the dictionary, neurotransmitters are associated to synapses. You need the chemical way to bridge a synapse between two neurons. And plants have no synapses, have no neurons or synapses. So if the dictionary says that you need a synapse, then you cannot be using it for signaling purposes. You see how stubborn and how short-sighted we can be. So instead of saying, hey, let's revise our terminology and do it more comprehensive and encompassing, such that any form of life can pass the test, provided that we customize and do the protocols and the experiments with an eye or thinking about what the plant cares about. But if we are not able to put ourselves in the shoes of the plant, it's impossible to interpret the results in a way that is consistent with the hypothesis that plants are conscious, are sentient, or that plants are intelligent, because we are trying to cast them into an animal mold. And that is the very mistake we have to avoid. In a sense, as we get started in Planta Sapiens in the preface, is say we need to avoid the navel-gazing attitude. Neither human or non-human animals can provide the gold standard of anything plants can do. So let's think about plant intelligence as an open empirical question. I suggest let's be self-critical. Let's try not to fall prey of biases, but let's do it also in a way that we don't avoid or prevent us from seeing potential plant smart behaviors, and we fail to recognize them because we try not to beef up or inflate plant behavior to avoid anthropomorphizing them. So we might risk anthropomorphizing plants, but we might also risk failing to see their intelligent behaviors when we try not to anthropomorphize them. You see what I mean? In a way, what I'm trying to say is that we need to train our eye to truly distinguish when we are looking at triangles and circles and when we are looking at plant behavior, that at its own time scale, we should be able to appreciate, provided we have the appropriate training and mindset. So I think I can leave it there, and then probably some of these topics we can elaborate in the Q&A or during discussion.
Thank you so much, Paco, for that lively and critical and yet very generous conversation, and such wonderful videos that you included as well. Those were fun, I think, for all of us to watch. I'm certainly full of questions. I don't know, Natalia, do you want to kick it off? Or
Well, I also am full of questions.
You're surrounded by plants. Let's see. Watch out.
Really, they're everywhere.
it's beautiful plants, yeah.
Thank you. We have a lovely relationship, me and my neighbors. I do have a question that I think could launch us into a discussion that comes from our recent reading group reading. So Rachel and I co-lead a plant consciousness reading group, or Thinking with Plants and Fungi reading group. We've been hosting it for the last 2 years at the Center for the Study of World Religions. And we recently read Pamela Lyon's piece. The title of the paper is "Of What is Minimal Cognition, the Half-Baked Version." And one of the things that she comments, which I think you spoke to, or you did speak to in your presentation, is that she, quote, end quote, "To say there is a minimal version of something is also to suggest that there is sufficient agreement about what the full-blown something is, such that we may recognize it when we see it. Concerning cognition, we have no such thing."
Absolutely.
So I'm curious, with the choice of the name of your lab, and also as you're speaking with both skeptics and people who are
Sure,
overinterpreting
sure.
results, I'm wondering how you're working with the term minimal in
Mm,
regards to cognition, and how you parse out the
mm.
complexity of
Yeah,
how we, as you said, like it's so much of it is about language and how we're speaking about things, and the
absolutely. Yeah, no, thanks. That's a great question to get started with. In fact, when I did, to be honest, when I started with the lab and just for whatever reason I came up with this name, I hadn't thought at the time about that issue. Then somebody once mentioned to me the very same thing, like, hey, why minimal, right? I mean, and I realized it was not the best choice of term for the name of the lab. Then I felt sorry about, I wanted to keep it. This is the IKEA effect. Oh, my little baby, my lab. I felt sorry for, I didn't want to get rid of the name. Then I came with a plan B, which is to say, no, minimal is our intelligence, not theirs, insofar as we fail to appreciate their smarts. So now, seriously, I'm on Pamela Lyons. I mean, we're on the same page. I fully agree with that, the point you are raising. In jest, I would say, hey, watch out who has minimal intelligence. But if you go, it doesn't need to be plants. If you go for bacterial cognition, protists, unicellulars, multicellulars, eukaryotes, any form of, that's why I say organism, any form of life in the tree of life as such. What we are after, in a sense, is identifying what unite us all in the tree of life. And that means that what unites us all, in my case, I think, at least my hunch, is that it has to do with integration. So the bacteria has to integrate all these cues coming from all these channels and provide, again, as in the case of the plant, a response that is globally adaptive. And we are talking about the unicellular. So we need to get rid of those gold standards. So plants or fungi cannot be a gold standard for each other or for unicellulars. So that's absolutely the case. So I would say let's just think of whichever organism we are is our model that we are trying to study, and forget about what the rest are able to do. Now, if you are gonna be able to find commonalities, you cannot ignore them. But not ignoring them doesn't mean to say that they've got to provide the metric. So comparative psychology is a great tool, but comparative psychology doesn't need to say that one of the species of the models we are comparing against the other is providing any gold standard. So yeah, fully agree with you.
Thank you. I love that. That makes me think of something Rachel and I have said a lot, which is rather than a hierarchy of minds or intelligences, thinking of it as a field or a plateau or a
Oh,
rhizome.
absolutely,
Yeah.
absolutely. Well,
Of,
think of the evolution of the eye. I mean, if you think of the evolution of the eye, the eye has evolved so many times independently, right? Then when we have, so imagine, this is a great example. I love this example. Imagine you landed on this planet where you only find vertebrates. So with camera type eyes, the type of eyes we have. And you haven't come across any insect, any invertebrate at all. And you're, scouting the surroundings, and you only encounter vertebrates. And you see them all navigating their surroundings visually. And you see they all have camera type eyes. And you infer camera type eyes are needed for vision. You couldn't see if you didn't have camera type eyes. And then the first time you come across one insect with compound eyes, so concave versus convex, then you should, if you were following that rule of thumb, you could say, oh, this insect cannot see. It's got to be exploring some other sensory channel, maybe hearing, smell, whatever. But it cannot possibly see because you need camera type eyes for seeing. That's what these skeptics were doing with respect to glutamate or GABA as a neurotransmitter. It cannot be because you don't have synapses. Then once you are open enough to say, hey, you don't need a camera type eye, you can have compound eyes like insects do, then you only need to realize that seeing through compound eyes, courtesy of compound eyes, or seeing courtesy of camera type eyes, is neither better nor worse. Is the type of eye that you need for the type of needs that you have. So we need to think of the type of needs that plants have. That's it. To put ourselves in their shoes rather than forcing them into an animal mold.
Relatedly, and continuing this conversation on language, you just spoke to this a little bit, but you've been part of this movement to really claim this field of plant neurobiology. But as you've illustrated, neurobiology itself is already indexed to animal-centric perspectives and to animal studies. And it seems like it's by the plant studies world, or a subset of it, choosing to claim plant neurobiology, you're kind of setting yourself up to fail, or constantly having to do this thing where you're metaphorizing, or people think you're metaphorizing. So yeah, what are some of the risks and opportunities of really claiming neurobiology as a plant relevant category? Like, how do you experience that?
Yeah, no, that's a great question again, because sometimes we think we are simply fighting over labels. And that's, if it was just fighting over labels, I would say, I would give it up. I would say, okay, what do you want to call it? I care about the actual over behavior I'm observing. You name it. You name it, but let me study the behavior, right? You know what the problem is? That look, I'm going to give you an example. Melatonin. The hormone, as we know it, you know, if instead of doing this on Zoom, I had crossed the pond to get there, I would be jet lagged. And I would have taken some melatonin, synthetic melatonin, right, for the jet lag. And melatonin, as we know it, the pineal gland secretes it. We know about that in animals since the 50s or 60s. And we only came across phytomelatonin in the 90s. And in fact, it took us almost a decade, from 95 to 2003 or something, for the scientific community to agree that that thing that you were seeing was melatonin, because it was not expected to be biosynthesized by a non-neural system. So plants were not expected to have melatonin. That was an animal thing, right? So that prevents, labels. So sometimes our expectations, they don't let us see things when they are just in front of us. You are seeing it and you still fail to see it because of the way it's being coached, right? Now, imagine how stubborn we are, that even when we agree that plants do produce their own melatonin, we find the need to stick in the prefix phyto. As in, no, this is, the real thing is our melatonin, they have phytomelatonin. It's the same molecule. It's the same molecule, for God's sake, right? Now, that's not even the problem. That's not even what worries me. You know what worries me? That by renouncing to the label, imagine we renounce to the label, we fail to appreciate the commonalities being shared across the tree of life that I was speaking about before. So if we don't see plant melatonin as on the same footing as animal melatonin, we might not even consider the possibility that plants can be jet lagged when they cross the pond. No, I mean it literally. So we now have a project, a project proposal. We submitted the proposal and we'll see if it gets funded. But it's a project on inducing jet lag to plants and reducing jet lag through through exogenous melatonin, phytomelatonin. So if I flew to the States and I was carrying a pet with me, a cat or a dog, I bet any of you would think, oh, my poor dog, my poor cat is got to be like me, like feeling, dazed and confused. We are both jet lagged. But if you were carrying with you a potted plant, one of those Natalie has on the shelf, I bet you would never think that your potted plant is jet lagged. And if you were to say so, people might say, yeah, a way of speaking. No. We mean it literally, because we are thinking that the application of melatonin is relieving the type of stress And we are talking about circadian clocks. So we are talking about the physiology. And if you don't put those labels, don't make use of the same terms, you will fail to be able to make the predictions. Think that theories, hypotheses, and if you coach them linguistically, we are talking about getting from premises to conclusions when you're doing science. And you might fail to see the connections if your language is not able to catch up with the developments.
Mm-hmm. As you were speaking, I was thinking of another person we've read in our reading group, the unfortunately recently deceased primatologist Franz de Waal, who has a wonderful paper on anthropomorphism, but also what he calls anthropodenialism, right? So that we, there's all this concern over overinterpreting data, but there's also a risk, as you say, where we miss the things that we share with other species. And in fact, he's a proponent of what he calls heuristic anthropomorphism. So it's a natural thing to do, and can we do it in a way to help us pose hypotheses that we can test, right? But yeah, there's a risk of underinterpreting, I think, as well. And a risk.
Yeah, definitely. So that's why it's so important, these biases, that first, that we proceed piecemeal and on a one-to-one basis. You know why? Because sometimes, I mean, there is no silver bullet. Sometimes you might encounter some behavior that, sure, I mean, you can think in terms of Morgan Cannon and say, hey, nothing much. Don't get overexcited. Okay, there is a way simpler explanation. That's it. But that doesn't mean that all plant behavior has to be either full or empty of cognitive interpretations, right? So sometimes you might find that it is the case that it had to be beefed up. Otherwise, we might be missing how Cuscuta was truly choosing between tomato and wheat. And some other times you might say, no, hold on a sec. I mean, don't take it easy, chill out. This was just reactive behavior. The plant simply grew towards the left and eventually reached the hole or bump onto the pole. It wasn't trying to get there. But sometimes you might be seeing intentional behavior. Sometimes it might not be intentional behavior. And some plant species might be doing one thing and other plant species might be doing something else. So we need to do it on a one-to-one basis, but not just for plants, also for animals, because it happens in our own case. In many cases, we beef up a lot of our cognitive abilities, and then we're like, hey, hold on a sec, that we are not that smart, or our smarts don't require all this cognitive machinery. Many times you can deflate them and say, hey, this can be explained, by outsourcing computational efforts into the periphery of the system, the body constraints or the environment, embodied cognitive science, all these things. And it's not about the recipe being followed in your head. It had to do with bodily constraints and morphological and material properties of your very body and the ground you're stepping onto, right? So we can deflate or inflate any system whatsoever. We just need to make sure that whichever way we do, we can grant that the leap from the data we are gathering to the conclusions we're reaching is justified with any model whatsoever.
Do we want to, Natalia, do you have another question? Or do we want to turn to Q&A? What do you think?
I have a quick question about replicated science. And so one of the things that we've talked a lot about in the reading group and has come up in previous discussions is the role of relationship in research. So for example, how the relationship between a researcher and say a lab rat affects the results of the experiments.
Absolutely.
And so I'm wondering, with the work that you've done thus far in the lab, if you have experienced or seen any form of similar reactions within the plant behavior. So for example, one of the things that came up with Monica Gagliano's work was a question of, could some of those experiments have not been replicatable because of the relationship she had with plants? And perhaps there was a reaction on a pheromone level or on, I don't know, some yet to be discovered experience. And so I guess my question is, what is the role, or have you seen a role that relationship plays in the way that
Mm.
plants are
Yeah,
behaving in the studies?
absolutely. Oh, definitely, definitely. Look, we have a PhD candidate doing his thesis in the replication crisis in the plant sciences. So we've been working hard on replicating Galeano's experiment, and this 11 issues paper will give you a lot of information just to understand how deep this whole problem space runs. But again, if you think about it, things are way simpler than we think they are. Because when you speak of the relation, how that might affect things, here, in a sense, we might even need to deflate the way that relation bears or affects plant behavior. What I mean to say is that an easiest way to proceed is to automate the whole protocol. So full automation, right? And full automation, but simply because the relation, physically speaking, simply has to do with, hey, I'm walking into the growing chamber, I'm picking, see what's going on. Oh, light is coming in, vibrations, perfume I'm wearing, noise, drop in temperature, change in relative humidity. So you are affecting the environment of the plant. So if, in a sense, that's obvious because you are breathing, you are doing things around the lab, so you are part of the lab environment and you are affecting it as such. What I mean to say is that the easiest way to deal with this is to have full control with automated protocols and say, if everything is fully automated, that's what we've done. Now we are running those experiments fully automated. Then you just get there at the very end, remove the card and watch the clip and see everything the plant did from day one. But you don't walk in to administer the stimuli. You are fully removed from the setting. That way, whatever happens doesn't depend on you. There is a different, more interesting sense, because this has to do with doing science, with rigor. But there is a more interesting sense in which cognition doesn't belong to the individual. Cognition spreads, is something, is collective. So I cannot cognize in the void. And in a sense, if you're going to identify some pattern of intelligent behavior, say in the wild, in the open, not lab conditions, then you cannot possibly ignore what other guys are doing, what they are up to, because my meaning making depends on yours. Now this is a triangle of eye contact in between we three. I cannot isolate Paco and study Paco in isolation. So in that other sense, it's not that there is a risk that the relation might affect the results, is that you have to include it because it's part of my cognition, because my cognition extends out there and implies and incorporates other agents, not just non-biological environment, but also the biological environment. So it's got to be seen or read or interpreted both in the positive and in the negative, positively speaking, and with, again, control when it comes to the running of the experiments.
Thank you. That's really, really interesting. And also, I wonder how, I suppose that is something that people have tried to do in labs with non-human animals and trying to find ways to take themselves out, but often that creates sort of a false objectivity that occurs with the lack of relationship. I'm wondering how you feel just within that, if perhaps that then, like, if there's a reflexivity statement or something that feels like necessary in explaining like why something had to be automated. If there's like, I guess that means I just need to read.
But how you mean? I'm not sure I understood you.
Like a reflexivity statement explaining like the philosophical background behind the experiment, but then also how that part of that philosophical background is that everything is creating meaning out of the relationship, right? That the plants are reacting to how, like you said, if you were in a natural setting, the plant would be
Right.
reacting to its entire environment, and you're isolating the plants and isolating the
Sure,
experiment
sure.
to have to look for specific kinds of behavior.
Yeah.
So I'm wondering if you feel like that then, because it's because there's so much skepticism around plant behavior being taken seriously, as choice or as memory or XYZ, if then there's like a stronger need that for some form of like reflexivity, positing statement.
Okay, I see what you mean. Okay, definitely. You know why? Think of the two hexagons I showed, the cognitive hexagon and the plant science hexagon. That's why it's so important that the plant scientists understand that they've got to team up with the cognitive neuroscientists and the philosophers. And I mentioned explicitly the arts and the humanities. And you know why? Because we have, correct me if I'm wrong, but sometimes have this sort of complex relationship with respect to the sciences, like, oh, they are doing the real thing, and I can be empirically informed. If I'm an artist, I can do a work of art, do some artistic creation out of the inspiration that comes out of the lab or what I've been reading or whatever. And we don't see those two channels as reciprocally connected. We don't see how those works of art can affect the scientific approach to plant intelligence. And I think that is got to be understood as truly reciprocal. Now, I'm mentioning this, I open it up not just to the philosophical foundations, but also to the arts and the humanities and the social sciences more generally, because it's so important to understand that data doesn't make sense as such in isolation without a solid theoretical framework in place. And that theoretical framework can only come by incorporating the social sciences and the humanities. Otherwise, you are, the type of science that we like to call in the philosophy of science a stamp collection. You are collecting stamps. So we cannot be gathering data, collecting data, and then I'm piling data up. And at the end of the day, I say, hey, I have an explanation. No, you have nothing, because data cannot be interpreted. This is like the philosophy of science 101 or ABC of philosophy of science. But sadly, in many science departments, students are not being taught this ABC of the scientific method. And they end up pursuing a PhD simply following a protocol. And they don't know what they are doing because they don't have the wider theoretical framework. And they simply turn the collecting data, you're extracting. And then you don't really understand why you are doing that, even if you are doing it really well. So very exquisite, precise protocols. You don't know what you're doing. You don't understand what the big questions are. We cannot get to the big questions from the gathering of the data. We need to have the big questions in the first place. Otherwise, we cannot make the predictions and we cannot think of the alternative hypothesis. So that's a very important point, because sometimes we think we are testing a hypothesis, and we are not testing a hypothesis. We are testing a space of hypotheses. And unless we are able, and that comes again to the idea of not giving up the battle on the terminology, because sometimes if you give up the battle on the terminology, you are going to fail to see an alternative hypothesis that might be there to explain the same data you are collecting. So we, first of all, need to do our homework in terms of the cognitive sciences, the philosophical foundations, and the theoretical foundations. Otherwise, you are just collecting data, and it's wishful thinking.
That's so helpful.
This tells again about the need to slow down. So you cannot do fast science for so many reasons, but this is a very important one.
I'm going to start bringing in some of these questions we have, because we have several and they're wonderful. Related, actually, I think, to this point that Natalia made about the impact of relationship on experimental findings. There's a question here about, is there any discussion of ethical treatments in plants in research, or do you think that will continue, or will that be a theme in the future, especially as we start to think about things like pain, for example? Has that been a discussion? Will it be a discussion?
Yeah. Oh, yeah, definitely. I mean, there is an ongoing discussion as of today. So plant ethics is an emerging field of research. Here we had a couple of days ago a couple of plant ethicists speaking about that. Yeah, certainly question number one many people raise, when you are giving a talk on plant intelligence, they say, hey, I'm vegan, I'm vegetarian, now what, right? And there are many concerns. Now, point number one, you cannot deny the very possibility of plant sentience on the grounds that that's gonna make you feel uneasy, or that now what? So whether plants are sentient or not is a scientific question. It is an open empirical question. With the theoretical foundations, we said we need to put in place, but an empirical question. And that cannot depend on whether that makes you feel more comfortable when you go to bed in the evening. Whether you sleep tight or not cannot be part of what makes us think that plants are sentient or not. And this looks like a joke, but I know many people who fall prey of this. They say, no, plants cannot possibly be. Otherwise, we are doomed. Now what? And there is an easy way out of this. You know what it is? The easy way out is to think that ethics applies to individuals, not to categories. It is this individual here and now, the one that is suffering or the one that has a good life. This individual here and now. And I stress this point because taxonomy can be helpful, but taxonomies can be very detrimental. And when we parcel the tree of life and split it apart into, hey, animal kingdom, plants, we do all these partitions, we think that the question applies to the partitions as such. And then people say, hey, do you eat meat? Do you eat vegetables? As if it was the category that goes to my table. No, to my table, on my plate, it is an individual that went through a stress or not the category it belongs to, the individual. So you see, if we frame the question along these lines, regardless of your choices, you will be dealing, or you will be concerned, genuinely concerned, with inflicting or trying not to inflict stress or pain to a form of life that might be potentially sentient. But again, the individual, right? So there is a lot of discussion going on. If this, whoever raised this question, will easily find a lot of literature, recent literature. So the field is in the making, it's an emerging field. There is a lot to be said, but the classical recipes don't work anymore. And I'm thinking of, animal liberation, Peter Singer. If you are an utilitarian and you say, hey, let's maximize good stuff around, well, you are in trouble because plants make up, most of the planet. So you could maximize by not caring about, 1% of animals. So again, why don't we think that this comes in degrees? It's a matter of degree, not in kind, and that it is the individual here and now, the one I should be concerned about, and not the category, which is an abstraction.
Yeah, I think that was really well said, and it resonates with something we've discussed in our reading group. I think for many of us, our principal relationship to plants is one of eating. And so it feels like consumer choices, buying what we choose to eat, is like the principal site of an ethic, right? So your principal ethic around plants is like, do I eat it or do I not eat it? When in reality, and then it becomes this like inflated choice, when in reality, there have been countless indigenous and animist traditions for whom all beings are alive, all species are alive. So the question then becomes, as you're saying, not do I kill this or do I not kill it, but what is the quality of my relationship to this being? Like,
Absolutely.
do I know this being? Do I have a relationship? Do I care for it? Do I steward it? So that's been a really rich area of exploration for us.
The very idea of personhood, we are sort of obsessed with human personhood. But if you think about it, I mean, it applies at many different scales in nature. So the personhood, again, of collective agencies.
Absolutely. I'll pull a different question from a scientist. in the Q&A, and then I'll pass it off to Natalia. So there's a question about what criteria do you use when choosing a plant species as a study system? For example, why use peas? Would you expect different behavior between wild and cultivated plant species?
Oh, that's a great question, because it's raising actually two issues, not just one. So one issue is that, as in animal studies, we are being enslaved by our preferred models. The plant model per excellence is Arabidopsis, right? A tiny grass with, some physiology, but many other plants do crazy stuff compared to Arabidopsis. But it comes handy, when we choose one model, it's always because of our interests, in terms of reproduction, success rate in transplantation, this or that. Think of fruit flies or, any animal model. So it comes handy to work with one model or another. Unfortunately, that, constrains the type of behaviors to be observed potentially. So definitely, the more we scout around different species and different surroundings, the more richness we will potentially find. Now, a different issue, very relevant, very interesting, is the difference in between whether it's a domesticated or a wild exemplar I'm studying. Because we forget that we've been domesticating plants for 10,000 years. It's called agriculture. We've been sculpting them to our needs, right? And then when we see this, you saw one of our videos, that the climbing being growing and reaching for the pole, we forget that climbing being has been shaped by us through agriculture, because we've been putting a pole next to it for thousands of years, right? Just think of your little dog, your pet, and wolves. So we've been doing the same to plants. What happens here is that, just even in this case, this domesticated vine won't care much about what's going on in terms of the need to explore, because a pole is always there, right? So the expectation, my prediction, is that wild counterparts will perform, we will be able to observe patterns of behavior way richer. They will need to explore way more, because it's absolutely a priority to them to find a support before it's too late. They need to get out there and reach out through the canopy, right? So they've got to be smart. Nobody's gonna come and put a pole next to you, right? So not just that, but we've been shaping their morphology. The phenotype itself is gonna affect, the bodily constraints are gonna affect the way it grows. Because when you domesticate a plant, the internode length, the length in between nodes, gets shortened. So if it's like dwarfed. So if a plant has less stem in between nodes, that's a shortened stem, it will circumnutate, it will revolve in circles way narrower. So it will just be doing like this. But if you are longer, if you stretch and your internode length is way longer, because you are a wild plant, then you will woo. And this woo is going to allow you to sense way more information from the periphery. You are exploring way farther, reaching out farther, right? So you see, now sometimes the third part of the question, it was 3 parts, not 2, is, for example, in the learning experiment, why pea plants? Well, in this case, it's obvious, because we are replicating a study, the one by Galliano, and in fact, we are using the very same genotype. So actually, Monica provided me with the seeds to perform exactly the same experiment. I should say that the critical attitude towards the replication effort doesn't mean to say that I don't believe that plants are able to associate. What I'm trying to say is that the experiment doesn't allow you to infer so. And yet, I have good reasons to believe that plants can learn by association. Why? Think about it. If you grow slow, if the way you move around and you move and reach, you grow from point A to point B, I need to get there, and that takes time, because I'm a plant, slow plant, I've got to make sure that I know where I'm going. So you need to anticipate. You need to predict the future. And the planet, our home and the home of plants, is full of regularities. So things happen on a regular basis. So it would be stupid, it wouldn't be justified on evolutionary grounds not to pick those regularities, because they are highly informative, right? So you see, there are two issues here that are separate. Do we have good reasons to raise the question in the first place? We certainly do. Maybe on top of all the issues we raise in the 11 issues paper, maybe a further issue is, hey, maybe pea plants aren't able to do so, or domesticated pea plants aren't able to do so. Certainly, we might need to check out what other species are able to do, both domesticated and wild. So that's why I say that this is, fortunately, work in progress until retirement. I will retire probably hopefully in 20 years' time, still wondering whether plants are able to learn by association. But I don't want to do fast science. I want to do slow science.
That's great, thank you. And that leads well into our next question, which comes from a poet in our audience and someone who attends the reading group regularly, which is, if we accept for now that plants are participants in cognition, perhaps even cognitive ancestors who build and sustain our own cognitive pathways, I wonder, first, what lessons have you drawn from plants' methods of interacting with ecosystems? And I'm thinking of your slow science model as one of the many that I'm sure there are they are sources of inspiration. And two, how have these lessons of the plant's method of interacting with ecosystems impacted your own methods of interaction?
Okay, that's a difficult question, because you know what? Here there is an issue of what might be going on out there that you are simply missing because of the time scale. And I don't mean that you've got to speed them up and then you will see it. I mean that the time scale at some point, and spatio-temporal scale as well, right? The whole spatio-temporal scale goes way beyond what you are able to appreciate. So you won't get to see the pattern, and the pattern yet is there, right? So when you're speaking of the ecosystem as such, you might be, in principle, unable to see the full pattern simply because it's going to take, well, a few centuries, a few hundreds of thousands of years, a few whatever. You don't know the trend. We are just having a snapshot. And sometimes that doesn't let you see the trend, where it's going. Is it going downhill or uphill? At what speed? So it's very difficult to I would say, I find that extremely difficult to see how to how to make that transfer of whether there is any isomorphism in between, structural isomorphism in between that type of relations and the ones you can either replicate or think about in the lab, because those might not fit with one another simply due to those constraints. And we never know how much of the footage we are able to see, just a few frames.
Thank you. Rachel, do you have another question from the Q&A, or Paco, a question from the Q&A you would like to respond to?
We'll try to do two more. This one may be a can of worms, but can you speak more about the ontogenic dimensions of plant intelligence? This seems to be a realm that crosses multiple scales of cognition between at least cells and organisms. Thinking here of Michael Levin's work, does the ontogenic perspective complexify the very notion of cognition and the plurality of scales in which it potentially exists? And if so, can you say more about at what scale cognition can be said to exist?
Yeah, well, it certainly makes things way more complicated and way more exciting, because we are far too used to thinking, hey, we simply deny this is a hardwired response, but still we don't quite understand how deep the experience that the individual has gone through during its lifetime affects or has to do with the way we are going to interpret this specific pattern of behavior I'm seeing right now, right? And just a very clear example. We said that plants are able to sense risk. So to mimosa, when you just think that, hey, you touch it and it falls its leaves, we might think, why do you need to appeal to ontogeny to deal with this? Pretty hardwired, an adaptation that evolved for the sake of being less conspicuous to herbivores, and yet some plants, individuals, might find it way more urgent to fold or come cannot feel the need to do, because they might have more resources. There is not such a strong metabolic demand in doing so. So depending on the past history, you might change gear. Things might not be as you thought they had to be were you to follow this recipe by default. So it's so important to see how those two, the phylogenetic and entogenetic dimensions, connect with one another. So it's we tend to put it to polarize it, to say, hey, is it either in the genes or does it have to do with the? And no, I think we need to see them both as part of a continuum, right? That's very important. One example we can add here is the Venus flytrap. When you touch the inner hair, right? We know you've got to touch it twice for the trap to close. It's like a security measure, right? So touch me twice just to avoid false positives. Well, sometimes Venus shuts just when you trigger the hair just once. It depends on the local conditions and it depends on the needs. So sometimes you say, hey, I can't afford to wait to just make sure it wasn't a false positive. So depending on your needs, and that's the here and now, you might change gear. And that's why plant behavior, on top of being adaptive, is got to be flexible. And flexibility can only have to do with ontogeny.
Thank you. We're going to finish up with one last question from our audience, and this comes from another scientist. And the question is, you mentioned that we need to gather more evidence for plant cognition. It makes me wonder at what point plants will no longer need to prove themselves to be intelligent. Why should we assume intelligence is absent in the first place? Why is the burden of proof on scientists to demonstrate otherwise?
Yeah.
Perhaps this isn't, goes into the ontological questions and the
Yeah.
philosophy framing.
Sure. Well, in a sense, there is a healthy sense in which we should assume voluntarily the burden of proof. So it's good to have the burden of proof, but not in plants, in any system whatsoever, right? The answer to this question has to do with whether you get started from a biogenic approach, and where there is life, there is mind, right? So to speak, to put it bluntly. But if you get started from the bottom up, all the way down, and then say, hey, mind relates to life. And by life, I mean unicellulars. Having to integrate all these channels we were speaking of. And then if that is the case, from a biogenic point of view, then things make pretty much sense. But that doesn't mean that you have removed the burden of proof. It means that the way you get started is from the bottom up and not from the top down. You are removing the gold standard we provide by default. But that doesn't mean that you don't need the burden of proof. We need the burden of proof, not plant scientists, but any scientist whatsoever, regardless of the model, animal or non-animal, in the sense that, in fact, when you don't have enough evidence, doesn't need to be flimsy, just could be very robust evidence, but very little, right? Because it's a new field, it's in the making, it's an emerging field. And that's not bad, that's good news. I mean, we mentioned phytoacoustics. Well, plant acoustics, if you read the papers in plant acoustics, the papers are like 2023, 2024, in print, 2019. So the literature in plant acoustics, it reduces to the last 10 years. So that's great news because everything is yet to be done, but it allows you to understand that having the burden of proof is good. You can have both the burden of proof and, theoretically speaking, assume a biogenic standpoint and get rid of that gold standard we are obsessed with. So no navel gazing, but no relying on anecdotal data. And that means that revolutionary results require robust data. That's, again, going back to the fast versus slow science. If you have this result, why do you have to move to the next hot topic? Stay there. Repeat it. Why you did an experiment with 16, 20 subjects? You got it published? Congratulations. But publishing the paper is not what we are after. We are after understanding behavior. And that means that regardless of whether you get it published or not, regardless of the impact of the journal, we do want to do those experiments and make variations round the clock. And that's a healthy way of assuming that burden.
I think that's a beautiful note to end on. Thank you so much, Paco, for this whole talk. And thank you, Natalia, for co-convening this. I think you've left us all with this beautiful dialectic of generosity, as well as a critical perspective that I think we need more of today. We need more of that complexity, as well as this deep and honest call for more interdisciplinary and transdisciplinary collaboration, which is what we are all about in our initiatives. So thank you so much. You are the perfect person to be part of our series, and we look forward to continuing the conversation.
Well, thank you. Thank you for having me. Thank you to everyone in the audience for the interest in plants. And I really enjoyed it. It was fun.
Sponsor, Center for the Study of World Religions.
Copyright 2024, the President and Fellows of Harvard.